everyone, welcome back to our NeetBioPrep channel. Let's continue the cell unit of life. Today we are going to learn cell envelope and its modifications. Most of the prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of the tightly bounded three-layered structure. They are the outermost glycocolex, followed by the cell wall and then the plasma membrane. Although each layer of the envelope perform the distinct function, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of the difference in the cell envelopes and the manner in which they respond to the staining procedure developed by the Gram-Vis. Those that take the gram stain are called as a gram-positive, and the other that do not are called as a gram-negative bacteria. Glycocolex differs in the composition and thickness among the different bacteria. It could be a loose sheath called the slime layer in some, while in other it may be the thick and tough called the capsule. The cell wall determine the shape of the cell and provide the strong structural support to prevent the bacterium from bursting or collapsing. Plasma membrane is selectively permeable in nature and interacts with the outside world. This membrane is similar structural to that of the eukaryotics. A special membranous structure is the mesosome, which is formed by the extension of the plasma membrane into the cell. These extensions are in the form of the vesicles, tubules, formation, DNA replication, distribution of the daughter cells. They also help in the respiration, secretion process to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotic like cyanobacteria, there are other membranous extension into the cytoplasm called the chromatophores, which contain the pigments. Bacterial cells may be motile or non-motile. If motile, they have the thin filamentous extension from the cell wall called the flagella. Bacteria shows a range in the number and arrangement of the flagella. Bacterial flagella is composed of the three parts: filament, hook, and basal body. The filament is the longest portion and extends from the cell surface to the outside. Beside the flagella, villi and fimbriae are also the surface structure of the bacteria but do not play a role in motility. The villi are the elongated tubular structure made up of the special protein. The fimbriae are the small bristle-like fibrous spotting out of the cell. In some bacteria, they are known to help attach the bacteria to rocks in streams and also to the host tissues. Here is an explanation of the cell envelope and its modification. We all know that cell envelope is outermost layer of the prokaryotic cell, and its main function is to give the protection to the cell and shape and support. It has three layers. They are: outermost layer is a glycocolex, middle cell wall, and the innermost layer is a plasma membrane. Glycocolex is nothing but the protective coat, and it protects the cell from the bursting or collapsing. It is the outermost layer and made up of the proteins and sugar. It helps in the bacteria to stick to the surface, escape the immune system. There are two types of the glycocolex: capsule and slime layer. Capsule is thick, hard and tight and protects strongly. Slime layer is loose and thin and less protection compared to the capsule. Main function of the cell wall is to give the shape and support to the cell, and it is made up of the special substance which is known as the peptidoglycan. Peptidoglycan is made up of the mixed substance of the sugar and amino acid. Cell wall gives a shape and prevent the bacteria from bursting in different conditions. There are two different types of the bacteria based on the cell wall. They are gram-positive bacteria and gram-negative bacteria. Gram-positive bacteria is in thick cell wall and gram-negative bacteria has a thin cell wall. Plasma membrane is the innermost layer of the cell envelope, and it is made up of the lipids, mainly bilayer. That mainly bilayer composed of the fats and lipids, and plasma membrane control the entry and exit of the material, and it is known as a semi-permeable, which allows some molecules or ions. Modifications of the cell envelope. They are mesosomes, fimbriae and pili and flagella. Mesosomes is formed by the extension of the plasma membrane and their extension in the form of the vesicles, tubules and lamina. We all know that. And mesosome help in the infoldings of the plasma membrane. And also main function is of the mesosome is to help in the respiration, DNA replication, cell wall formation, distribution of the daughter cells, secretion process, increase the surface area of the plasma membrane and enzymatic content. Fimbriae is the small hair-like structure on the surface and help in attachment to the surface or other cells. Pili are also used in the gene transfer, which is known as a conjugation. Flagella is a tail-like structure for the movement of the bacteria. Here is a summary points of cell envelope and its modification. Cell envelope is composed of the three layers. We all know that glycocalyx, cell wall and plasma membrane. All these three together called as a cell envelope. Glycocalyx is a protective layer and it's contain the two different types of the layers. They are capsules and slime layer. We all know that capsule is thick and slime layer is a thin layer. Cell wall main function is to give the shape and support to the cell, and it is made up of the peptidoglycan, which is mixed substance of the sugar and amino acid. Plasma membrane control the movement in and out of the molecule. Then it is called as a semi-permeable in nature. Modifications of the cell envelope and its modifications: mesosomes, fimbriae and pili and flagella. Ribosomes and inclusion bodies. In prokaryotic, ribosomes are associated with the plasma membrane of the cell. They are about 15 nanometer by 20 nanometer in size and are made up of two subunits, 50S and 30S units, which when present together form the 70S prokaryotic ribosomes. Ribosomes are the site of the protein synthesis. Several ribosomes may attach to the single mRNA and form a chain called the polyribosome or the polysome. The ribosome of the polysome translate the mRNA into the proteins. Inclusion bodies. Reserve material in the prokaryotic cells are stored in the cytoplasm in the form of the inclusion bodies. They are not bounded by any membrane system and lie free in the cytoplasm. Example: phosphate granules, synophiaceae granules, glycogen granules. Gas vacuoles are found in blue, green and purple photosynthetic bacteria. Ribosomes is nothing but the protein factory because it only synthesizes proteins. Ribosomes are the tiny rounded structure found in both prokaryotic and eukaryotic. Except a ribosome, nothing can be seen in both eukaryotic and prokaryotic. Only ribosomes are present in both eukaryotic and prokaryotic. The main function of the ribosomes are to synthesize the proteins. They are made up of the RNA and proteins. In prokaryotic, ribosomes are about 70S and two subunits are 50S and 30S together form the 70S ribosomes. In the eukaryotic, the ribosomes are the 60S and 40S together form the 80S ribosomes. Ribosomes can be free in the cytoplasm or attached to the endoplasmic reticulum only on the rough endoplasmic reticulum. Due to the presence of the ribosomes and the endoplasmic reticulum, it gives a rough appearance. Then it is called as a rough endoplasmic reticulum. Free ribosomes make proteins used inside the cell, while rough endoplasmic reticulum make protein to send out of the cell. Ribosome is nothing but the protein making machine. Inclusion body is also known as a storage bags, and it is only found in the prokaryotic, not in the eukaryotic like ribosomes. These are non-living structure used to store waste and food material. They do not have membrane, life free in the cytoplasm. Examples of the inclusion bodies are glycogen granules, phosphate granules and sulfur granules. Here are the few questions based on the cell envelope and its modification. Ribosomes and inclusion bodies. Question number 1. Which of the following currently represent the layer of cell envelope in prokaryotics from the outside to innermost? We all know that the outermost layer is a glycocalyx, then followed by the cell wall, and the innermost layer is a plasma membrane. Option B. Question number 2. Slime layer in the bacterial cell envelope helps in. We all know that glycocalyx is composed of the two layers, capsule and slime layer. They both helps in the protection. Option B. Ribosomes in the prokaryotics are of which type? Prokaryotic have 70S and ribosomes in the prokaryotic are of which type? We all know that prokaryotic ribosomes are about 70S, which is made up of the two subunits 50S and 30S. Eukaryotic ribosomes are the 80S, consist of 60S and 40S. Option A. Fourth question. Inclusion bodies in prokaryotic cells are also known as. We all know that inclusion body is also known as a storage bags and it store the reserve material. Option C.